Welcome, everyone. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. We invite you to put a note in the chat introducing yourself if you'd like to. I'm going to start off with a few bits of housekeeping. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of environmental health sciences, the Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. If you're joining by phone, you can download the slides from the webinar page on our website at healthandenvironment.org. And for those who are online, we'll put that link in the chat now. We'll disable the chat during the presentation, but please do type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. If you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. This webinar is being recorded, and it's scheduled to last for half an hour, though we might go a few minutes over depending on the questions. I now have the pleasure of introducing today's speaker. Dr. Mark Purdue is a senior investigator in the Occupational and Environmental Epidemiology branch of the National Cancer Institute's Division of Cancer Epidemiology and Genetics. Dr. Purdue's interests center on applying molecular and classical epidemiologic methods to identify environmental and occupational risk factors of cancer. He is particularly interested in evaluating the potential carcinogenicity of PFAS and of chlorinated solvents, and in investigating the etiology of kidney cancer. We're really glad that Dr. Purdue is able to join us today to discuss his recent study on PFAS and testicular germ cell tumors among U.S. Air Force servicemen. Dr. Purdue, thanks so much for joining us today, and the floor is yours.
Thank you, Rachel. And it's a pleasure to be with you today and to have the opportunity to share with you findings from this study that my colleagues and I conducted to investigate the relationship between serum PFAS concentrations and testicular cancer among U.S. Air Force servicemen. So just starting with a disclaimer. So just for an outline of today's presentation, I'll begin by providing a background on PFAS before moving into a description of our study and our study findings. And then I will close with a summary of what I've discussed and offer some possible next steps in research in this area. So PFAS refer to per and polyfluoroalkyl substances, which are a class of man-made chemicals involving fluorine atoms attached to carbon chains of varying length and complexity. PFAS have some unique properties imparted through these fluorine carbon bonds that lend themselves to be quite useful commercially. Some of these properties include imparting oil and water repellency, as well as resistance to high temperatures and a reduction in friction. The first PFAS was invented in the 1930s, and within a decade, companies began manufacturing PFAS for use in a variety of commercial products. In particular, PFAS have been used for nonstick coatings for products such as cookware, textiles and fabrics, and paper products, food wrappers in particular. PFAS have also been used in specialized firefighting foams that are used to extinguish flammable liquid fires. These are known as aqueous film-forming foams. And PFAS are a key ingredient such that when these foams are applied to a liquid, the PFAS help the foam disperse rapidly in a very thin layer above the liquid to serve as a barrier, starving the fire of oxygen. The Department of Defense has been using these aqueous film-forming foams for many decades, dating back to the late 1960s. Typically, the The firefighting foam formulations used by DoD and contained the chemicals PFOS, perfluorooctane sulfonic acid, perfluorohexane sulfonic acid, or PFHXS, and sometimes perfluorooctanoic acid, or PFOA. Although in more recent years, they've moved away to different types of foams. And so in particular, PFOS, which was the chemical used predominantly in the firefighting foams used by DoD, PFOS was phased out of production in 2003, roughly, due to concerns over its potential health effects. And thus, by extension, PFOS-based firefighting foams were no longer being manufactured, although DoD continued using its existing stockpile of firefighting foams for many years. In 2016, DoD introduced new rules restricting the use of PFOS-based firefighting foams, and it also initiated systematic water testing across all military water systems employed at installations. From this testing initiative, DoD identified 401 bases with known or suspected PFAS releases, as well as 90 bases where groundwater samples had measured levels of PFOS and PFOA exceeding 70 parts per trillion, which was the EPA lifetime health advisory at the time. As well, 36 bases were found to have drinking water samples with levels of PFOS and PFOA exceeding that 70 parts per trillion threshold. And more recently, DoD began a conversion to firefighting foams using newer short-chain PFAS. So these are different types of PFAS that are thought to be have a safer profile. And they're also exploring alternative foams that are PFAS-free. So although many of these PFAS, like PFOS in particular, have been phased out of use, they're still of concern as potential health risks. And that's due to the fact that these chemicals are highly resistant to degradation and persist in the environment for a very long time, which has led to their nickname as forever chemicals. And these days, PFAS are now detectable in the majority of Americans in blood tests. PFAS can be, exposures can come from a variety of sources. A small number of individuals can have occupational exposures to PFAS, but the majority of individuals are exposed through consumption of contaminated food and, in particular, water. And it's important to note that AFFF foams used at commercial airports and military facilities has been identified as an important source of how PFAS gets introduced into public water systems. And PFAS are also suspected to have some health effects. Epidemiologic studies have suggested associations with a variety of different types of diseases and conditions, although a few for which the evidence is more consistent include associations with elevated cholesterol, an elevated risk of pregnancy-induced hypertension, as well. Some PFAS have been shown to induce immune effects, to affect thyroid function, and to be associated with risks of some cancers. Regarding PFAS and cancer, in 2014, the World Health Organization's International Agency for Research on Cancer conducted its first monograph evaluation of a PFAS chemicals to evaluate scientific evidence as to whether the level of evidence that this agent may be a human carcinogen. And so in this initial evaluation conducted by IARC, they focused on the chemical PFOA. From that evaluation, they classified PFOA as a possible human carcinogen, noting the human evidence to be strongest for associations with cancers of the kidney and testis. Fewer studies have investigated cancer associations with other types of PFAS, including PFOS. And thus far, the evidence is largely inconclusive, although it's worth noting that, in fact, IARC has a follow-up monograph review ongoing as we speak, conducting a reevaluation of PFOA with the updated evidence, scientific evidence to date, and it's also evaluating for the first time the evidence for PFOS. So it's interesting that although PFAS releases through AFFF use at military bases has been identified as an important source of exposure, and there's been a lot of work focusing on potential exposures and health risks among communities neighboring military bases. There's actually been very little work done to evaluate the levels of PFAS exposure among military personnel themselves. And so it's been unclear what kinds of PFAS serum burdens exist among military personnel and how those PFAS levels compare to the general population. As well, it's unclear whether there exist service-related predictors of elevated PFAS exposure. And also it's not known at this point whether PFAS exposures are associated with cancer risk in military populations. So to address these knowledge gaps, my collaborator, Dr. Jennifer Ruszecki at the Uniformed Services University, and I, we organized an epidemiologic study to investigate the relationship between serum concentrations of PFAS and risk of testicular cancer in Air Force servicemen. So why testicular cancer? Couple of reasons. Firstly, it's the most commonly diagnosed cancer among active duty servicemen. And that's a direct consequence of testicular being a cancer of young adult men. And that's the age group at which it's most commonly diagnosed. However, we were also interested in testicular cancer given previous evidence suggesting associations with PFOA. And we focused on Air Force personnel, given the findings from that DoD testing of water samples from military drinking water systems. From that initiative, half of the bases that were found to have known or suspected PFAS releases were Air Force installations. So just a few details about the study design. Our source population was active duty Air Force servicemen with serum banked within the DoD Serum Repository, involving serum collected between 1988 and 2017. We linked records from the DoD Serum Repository to the Military Cancer Registry to identify 530 cases of testicular cancer diagnosed during active duty, and for whom, among whom there were pre-diagnostic serum samples within the repository. And then we individually matched one control to every case on the basis of the matching factors listed there. For most subjects, we utilized only one banked serum sample from the repository per subject. However, we did use 2 samples per subject for cases diagnosed at least 5 years after the earliest available collected serum in the repository, and as well for their matched controls. So there were 187 such case control pairs. And the first sample that we selected was usually collected around the start of military service, and the second sample was usually collected several years later. And then we worked with our collaborator, Dr. Antonia Califat at the Centers for Disease Control and Prevention, whose laboratory received these serum samples and measured concentrations of 7 PFAS using their mass spectrometry protocol. And so on to the results. Just briefly, I want to give an overview of some characteristics of the study participants. A majority of the participants were less than 35 years of age as of the case diagnosis date. The majority were non-Hispanic white and were enlisted personnel. So the first thing we wanted to do in our analysis was to compare the mean levels of measured PFAS from among the controls among our Air Force servicemen to mean levels of PFAS measured within some national general population surveys, specifically the National Health and Nutrition Examination Surveys. So this slide summarizes the geometric mean concentration of PFOS for Air Force controls and for NHANES samples. We're making these comparisons in relation to the years of specimen collection from the corresponding sources. And you can see here from this slide that in both populations, we observed a sharp drop in levels of PFOS in the blood. And this is not surprising. It's been observed in the literature and is a consequence of PFOS being phased out of production in the early 2000s. In comparing the levels between Air Force controls and NHANES subjects, we don't see really any notable differences in levels of PFOS. So this slide shows similar results for PFOA. Again, we see a decline over time, a weaker decline than PFOS, and this is consistent with the phase out of PFOA being more protracted and taking place at a later point in time than for PFOS. We see maybe some hints of higher levels of PFOA among Air Force controls versus NHANES subjects, although statistical tests were generally not statistically significant. This slide summarizes the geometric means for perfluorohexane sulfonate. And in this slide, we can see higher levels of this chemical in the serum of Air Force controls compared to NHANES subjects for samples collected prior to 2004. So moving on, another aspect of our analysis involved exploring whether there existed service-related predictors of elevated serum PFAS concentrations from among the service-related data that we had available to us. We identified 2 such predictors. The strongest predictor was employment in firefighting as a military occupation. And we observed military firefighters to have substantially higher levels of PFOS, PFOA, and perfluorohexane sulfonate versus other servicemen. We also observed weaker evidence suggesting elevated levels of perfluorohexane sulfonate among individuals who prior to the serum collection had been stationed at an Air Force installation where drinking water samples had been found to have elevated concentrations of PFOS and PFOA exceeding the 70 parts per trillion EPA lifetime health advisory at the time. So then we move on to case control analyses, comparing levels of PFAS in cases and controls. And for those of you in the audience who may not be familiar with these kinds of statistics, we're using odds ratios to evaluate associations. And just to give you a sense of how to interpret an odds ratio statistic, an odds ratio greater than 1 would indicate a higher frequency of a particular exposure among cases compared to controls. So this slide summarizes odds ratios from categories of measured serum PFOS concentration from the overall set of study participants. And in this initial analysis, we did not observe an association with PFOS, although when we did adjust in our regression models for other PFAS, we did observe evidence of a weak suggestive association that did not reach statistical significance. We moved then to analyses within the subset of participants that had 2 samples. And in our analyses of measurements from the first samples, we observed findings very similar to what we observed overall, with evidence of a weak positive association that was non-significant. Although when we analyzed measurements from the second samples, we observed stronger evidence suggesting an association with an exposure-response relationship that reached with a test of trend that reached statistical significance. And when we adjusted our models for levels of other PFAS, these findings remained, and if anything, were slightly stronger. We did not observe evidence suggesting positive associations with testicular cancer for the other measured PFAS, and that includes PFOA. Although it's worth noting that the PFOA levels in our study are very comparable to those, I would say, in the general population. And many of the previous studies that have suggested an association with testicular cancer and PFOA involved study populations with much higher exposure levels to PFOA. So I think that's important to consider when interpreting our results. We can't rule out an association at higher exposure levels because we didn't really see those kinds of exposure levels within this study. We also observed evidence of an inverse association with testicular cancer for serum levels of perfluoro non-anoic acid, although it's worth noting that the association was largely confined to cases diagnosed within 5 years after serum collection, which would not be compatible with an etiologic relationship. So to summarize our key findings, in general, we observed fairly similar levels of serum PFAS among the Air Force controls compared to the NHANES samples representative of the general population, with the exception of higher levels of perfluorohexane sulfonate in Air Force controls for samples collected in the early 2000s. We identified 2 service-related predictors of elevated serum PFAS. Certainly the clearest predictor was occupation as a military firefighter, as well as suggestive evidence of prior service on a base with drinking water samples found to have high PFAS levels, an additional possible predictor. And in our case control analysis, we found evidence suggesting that elevated serum PFOS is associated with increased risk of testicular cancer. And as I mentioned, we observed null findings for PFOA. So these findings are, to our knowledge, the first evidence of a direct association between measured serum PFOS concentrations and testicular cancer. It's unclear what mechanisms may underlie this association. Although it's worth noting that it's been hypothesized that endocrine disrupting chemicals may contribute to the pathogenesis of testicular cancer. And in that regard, there is experimental evidence from several studies in animal models suggesting PFOS-induced male reproductive toxicity. So just some important research questions, I think, that follow from these findings is firstly, the question of whether these findings are replicable. And that's something that my collaborator, Dr. Rishecki, and I are hoping to address in a follow-up study that we're organizing within the DoD Serum Repository, this time using samples from servicemen in other military branches. It's also worth noting that the majority of serum samples that we used in our study had been collected in the 1990s and 2000s. And it's not clear whether what PFAS serum levels are among current DoD personnel from the findings in our study. And so I think that's an important question that warrants investigation, as does the broader question of whether PFAS are associated with other health conditions among military personnel and veterans. And lastly, another important question for further research is that of whether short-chain PFAS are safe. Now, these are PFAS that are now being used in AFFF foams and in some other applications as substitutes for these original longer-chain PFAS, for which we have evidence suggesting health risks. These short-chain PFAS, as I may have mentioned earlier, are cleared from the body faster, have shorter half-lives in the environment, so they're less persistent. But I think there's still insufficient evidence at this point as to whether they exert any toxic effects, either in animal models or in humans. And I think certainly more epidemiologic evidence of these newer classes of PFAS are needed. So with that, I'd like to close by acknowledging my study collaborators and funding sources, and in particular, Dr. Rishecki, my co-PI on this project. And I'd be happy to take any questions you may have. Thank you.
Great. Thank you so much. That was an excellent presentation. So I will just start with a question from Dick Clapp, which is, could you also look at PFAS and kidney cancer in this cohort?
The short answer is yes, although it's worth noting that the current infrastructure for linking the military cancer registry to the DoD Serum Repository has the limitation that the military cancer registry essentially is capturing cancer diagnoses among active duty personnel, as well as beneficiaries covered under the same military health insurance and retirees from DoD. So these are people with 20 years of service who are eligible for TRICARE benefits. It does not include, or it does not systematically capture cancer diagnoses among veterans at large. And kidney cancer is a cancer of older adults. And so if we're only focusing on active duty military personnel, there are fewer kidney cancers diagnosed within that population than among veterans, for whom we can't readily link to the serum repository. But that said, yes, absolutely, we could study kidney cancer, and it is feasible to study that. We just won't be capturing the kind of optimal age range when we focus on active duty personnel.
Got it. Thank you. That's a really helpful answer. The next question that I see here is a question asking you just to expand a little bit on routes of exposure to PFAS. And I'll just add to that and ask if somebody believes or knows that they have had significant PFAS exposures, are there particular recommendations that you would have for them?
So regarding the first part, I'm not quite sure what I'm being asked to expand on. Except I guess I would say that the majority of the general population are likely exposed to PFAS through consumption of water with PFAS contamination, Diet is also recognized as a source of PFAS exposure. Some PFAS have been found to bioaccumulate, not all, but some types of dietary intake have been linked to elevated PFAS, including in some populations fish intake and meat intake. I think there's still more research to be done as to whether exactly what dietary sources are important with regard to PFAS exposure. Regarding the question of what if someone's concerned that they may have had exposures to PFAS, what should they do? I would say, well, firstly, with the caveat that our findings, we need to replicate our findings. So I think it's important not to draw strong conclusions regarding causation at this point. But certainly there's reason to be concerned about PFAS in general. And if individuals think they may have had exposures to these chemicals and are concerned, I would recommend that they talk to a physician. I would also note that the National Academies of Science, Engineering, and Medicine a couple of years ago published a report offering guidance to physicians regarding how to handle patients with concerns over exposure, with regards to what circumstances where testing for blood PFAS levels may be warranted, and what to do in the way of workup or a follow-up of patients in those scenarios. So I would suggest they talk to a physician if they're concerned.
Great, thank you. So we're a little bit over the half-hour mark, but I'd like to bring up at least one, maybe two more questions. So we have a question about whether you could study associations between exposures and cancers among military family members. And there's also a question about whether you could look at cancer in women, including ovarian cancer. So just anything there that you're able to comment on.
Yeah, well, certainly, I think it's important to study exposures among women as well. Our group recently published findings suggesting that elevated serum levels of PFOS are associated with an elevated risk of ER and PR positive breast cancer, which is a finding that's consistent with a smaller, earlier study. And I'm aware of other ongoing research focusing on ovarian and endometrial cancer, and it'll be interesting to see when those findings, when they're published. And as well, as I mentioned, the International Agency for Research on Cancer is currently conducting a reevaluation of the evidence surrounding PFOA and its first evaluation for PFOS. And they will publish a monograph as well as a scientific published paper summarizing their findings from that review of the literature. And that'll offer important guidance to scientists, moving forward. But certainly, studying female reproductive cancers is an important area for investigation. And looping back to the original question around families of military service personnel, it's certainly it's possible insofar as if those individuals receive TRICARE benefits, that there's potentially the groundwork in place to conduct studies of those family members. I guess the important question is whether family members have elevated serum PFAS concentrations. And I'm not aware of any data suggesting that, and that may be an important area for further investigation. Yeah, thank you for that suggestion.
Great, thank you. Well, I think I'm gonna mention one last question if you just want to give a quick answer, and then we'll wrap up since we're over time. So this last question I'll just read is asking, what do we know about duration of exposure to PFAS and risk? And this person asks, presumably serum levels could reflect a 1 or 2 time acute exposure and or a long-term exposure.
Yeah, that's a good question. So when you're using, and you want a short answer, the short answer is, yeah, it's serum measurements are don't offer the complete picture with regard to someone's exposure, and it often depends on how rapidly the chemical of interest is cleared from the body. If it's something that is in and out of the body within days or hours, it's going to be a biomarker reflecting recent exposure. Some chemicals, such as PFAS, persist in the body for years. So the serum half-life for PFOS is approximately 2.5 to 3 years. So it is a better surrogate of longer-term exposure. But what you measure is going to be a reflection of the duration and the intensity of exposure. And the ideal study would measure blood levels, but also collect information on an individual's past history of, of what they ate and where they lived, and, where they got their water, so that they could try to reconstruct an idea of their cumulative exposure. Some studies are able to do that, but don't necessarily have the blood. In this case, we have the blood, but we're not able to reconstruct their history. So it's an important issue. And, I think moving forward, we want to try to design studies that can best capture an individual's exposure history, incorporating both elements.
Great. Thank you so much. I really appreciate your excellent answers to these questions, and thanks thanks to everyone for putting these excellent questions in the Q&A. So we will wrap up now. I'll just make a few quick announcements. A video recording will be available on CHE's website soon, and you will all receive an email with a link to the video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's Partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners. We also invite you to join us for upcoming webinars. On Wednesday, November 29th, the next CHE Alaska webinar will explore breast cancer pinkwashing and the importance of prevention. We'll be hearing from Nancy Buermeyer of Breast Cancer Prevention Partners and Crystal Redman of Breast Cancer Action, and I'll put that link into the chat in a moment. And then on Tuesday, December 12th, our next EDC Strategies Partnership webinar will focus on neuroendocrine disruptors and brain development, lessons from PCBs. We'll be hearing from Dr. Tom Zoller about how the endocrine disrupting effects of PCBs can affect human brain development, and making the links to broader implications for chemical testing and regulation. And again, I'll put that link in the chat in a moment. So again, we'd like to thank all of you for joining us today. Thanks so much to Dr. Purdue for your really important research and for taking the time to give this excellent presentation. And many thanks also to CHE Director Kristen Schaefer for her behind-the-scenes support.